Hello everyone, this is Dr. Hava Bekirolataş from Ministry of Health of Turkey and I am the Laboratory Manager of the Chemical Warfare Agents Diagnosis and Verification Laboratory. Today, I will present a laboratory study on the verification of VX, a highly toxic organophosphorus nerve agent, and VX-related compounds within the framework of an OPCW proficiency test. Let me briefly outline the structure of my talk. I will first give a very short introduction to chemical warfare agents and nerve agents, then I will focus on the reporting procedures in OPCW proficiency tests and show how VX and a VX-related degradation product were confirmed in our laboratory. Before going into details, I would like to briefly introduce our laboratory. Our lab is the only facility in Turkey that performs diagnosis and verification of chemical warfare agents, and it is also one of the OPCW designated laboratories worldwide. And this makes the results we generate highly significant both for our country and for the implementation of the Chemical Weapons Convention. Chemical warfare agents are chemicals produced to cause physical and psychological destructive effects in humans. They may also be used to contaminate plant and animal resources, or to render economic targets inoperative. They span a very broad chemical space with many agent families that show diverse physical and chemical properties. One of the most important families among them is organophosphorus nerve agents. Nerve agents can be divided into several subclasses, and within these, VX-type agents stand out as highly toxic and relatively persistent compounds. As their name suggests, nerve agents target the central and peripheral nervous system. They inhibit the enzyme acetylcholinesterase, causing the acetylcholine to accumulate at receptors. This leads to characteristic cholinergic signs and, depending on the level of exposure, can result in life-threatening and even fatal clinical outcomes. On this slide, you can see some examples of VX-type nerve agents. In the Schedules of Chemical Weapons Convention, they are listed under Schedule 1A03 and are characterized by a C3 alkyl group directly bound to the phosphorus atom. VX-type agents undergo degradation through environmental factors, hydrolysis, and decontamination processes, giving rise to various transformation products. In the PT scenario that forms the basis of this work, VX and series of VX-related degradation compounds were included, as you can see on the slide. Let me now briefly explain OPCW proficiency tests. In these demanding exercises, participating laboratories receive a total of 6 samples, that is, 2 sets of test items prepared according to a given scenario. The laboratories are expected to analyze the samples blindly for CWC-related chemicals and correctly report the spiking chemicals present. In OPCW PTs, the results are evaluated according to predefined reporting criteria. Laboratories are expected to confirm each reported chemical using at least 2 different spectrometric methods. And this is particularly important for critical agents in order to minimize both false positive and false negative findings. Now, let us look at the scenario of the 55th OPCW proficiency test. Participants were asked to analyze 2 types of samples described as solvent waste and decon waste, and the aim was to investigate the presence of VX and related degradation products in these waste streams in line with the given scenario. In this test, there were 8 spiking chemicals in total. Clearly, it's not possible to describe all of them in detail within a 10-minute talk. Therefore, today I will focus on VX in the liquid waste sample and show how it was reported based on GCMS analysis with a reference standard. And in addition, I selected a hydrolyzed product of VX with a toxicity close to that of VX and commonly known as EA2192 as an example from the decon waste sample, and it was reported by our laboratory without the use of a reference standard chemical. Okay, let us start with VX. I will show how VX was confirmed by GCEIMS and GCCIMS using a reference standard, and this will illustrate the reporting strategy when a suitable reference material is available in your lab. The sample preparation steps applied to the liquid waste are shown on the slide. Vx was detected in the liquid that was subjected to solvent exchange and concentration. In other words, careful pretreatment allowed us to enrich the target agents in a specific fraction. For the force method, we used a GCMS system operating in electron impact mode. And in OPCW-PTs, a typical sequence is to inject the solvent or method blank, followed by the sample and then the reference standard. And in the expected retention time window of the spiking chemical, the blank chromatogram must not show any peak, whereas the reference chromatogram must display a clear peak. And in this study, these conditions were fully met for both blank and reference injections. A comparison of the mass spectra is also a critical step. In general, a spectral similarity of at least 80% is required in OPCW-PTs, and for Vx in this test, the similarity between the acquired spectrum and the reference spectrum was about 95%, providing strong support for structural conformation. And confirmed the detected chemical by a second method, the same sample was also analyzed by GC chemical ionization MS using methane as the reagent gas. Again, the blank, the sample, and the reference were analyzed consecutively by CI, and in the relevant retention time window, no interfering peaks were observed in the blank, and consistent peaks were detected for both sample and reference injections. As you can see, the CI spectrum looks quite different from the I spectrum, which is fully consistent with the softer nature of CI. And here, the signal at M to Z 268 corresponds to the M plus H ion of Vx and matches well with its molecular weight, adding further confidence to the identification. The example from the Decon waste is, as mentioned, a hydrolysis product of Vx. Because of its higher polarity, this compound is not directly suitable for conventional GCMS analysis. Therefore, the spiking chemical was converted into a TBDMS derivative to increase its volatility and make it amenable to GC. And the analyte was then detected in the derivatized liquid. And the derivatized sample was analyzed again using GCMS in EI mode. And on this slide, you can see two chromatograms. The first one is the blank, and the second one corresponds to the TBDMS derivative of the spiking chemical. And no peak is observed at the target retention time in the blank, while a clear peak appears at the expected retention time in the derivatized sample. And the EI mass spectrum obtained for this peak was searched again in NIST database, yielding a similarity score of about 96%, and such high similarity is extremely valuable when no reference standard is available, as it strongly supports the proposed structure. And let us now briefly consider the CI results. And in this case, isobutane was used as the reagent gas. And in the CI chromatograms, we again observed a clean blank and a distinct peak for the target compound at around 21 minutes. When evaluated together with the EI data and the derivatization information, this retention time adds another layer of confidence to the identification. And in the CI mass spectrum, the signal at M to Z 354 corresponds to the M plus H ion of the TBDMS derivatized compound. And again, combining EI and CI data, the derivatization approach, and the library search, we were able to reliably identify this Vx hydrolysis product even in the absence of a reference material. According to the official OPCW evaluation, our laboratory obtained an A score in the 55th proficiency test, and this means that Vx and all Vx related compounds were reported correctly and in line with the scenario requirements. And this outcome was particularly important for us because following this test, our laboratory was officially granted OPCW designated laboratory status. And at present, there are around 30 OPCW designated laboratories worldwide, and one of them is located here in Turkey. And having this capacity in our country is a strategic advantage for the assessment of potential chemical incidents and for CWC-related verification activities. Thank you very much for your attention, and I will be happy to take your questions and comments.